and is very active. And you might have seen her yesterday also here on the floor. Then we have Uli Burger, who you have also seen yesterday here. He's a statistician at Roche. He's currently the president of BBS, and he's a member of the ICH E20 team, which is relevant for this session. We have Frank Breetz, who is a quantitative research scientist at Novartis. He's also a very active contributor to methodology, and he's also a member of the ICH E20 team. So please welcome these three. I understand that Uli and Frank will share the presentation. Over to you.
Good. This should be okay. Thanks, Kaspar. So I will start with a presentation. Frank will end, and Khadija is part of it. She worked a lot on the slides as well. Just to clarify, we are all three members of ICH E20, but this is not a report from ICH E20. Although what we will say is, of course, when you went through the process over many years with writing up a guideline, it will be influenced by what we discussed in E20. The first thing is that there are many different definitions of adaptive designs, and for our approach, it's important to define more clearly what we mean with adaptive design. And what is really with the definition here, what is really important is that we think an adaptive design is a clinical trial design where the prospectively planned modifications in the design and based on accumulating data from subjects in the trial, these are really the two key assumptions we make. Okay. And of course, adaptive designs, you can think about them in the exploratory and in the confirmatory framework. The confirmatory framework is certainly the more challenging one. Move it down. Okay, thanks. Why using adaptive designs? I think we are probably, most of us are pretty familiar with this. They can address uncertainties at the design stage when you plan a trial, on the target population, outcome variability, on the size of the trial. And adaptive designs enable, in principle, modifications of the trial design during the study. So they provide more flexibility and can, by that, really decrease time to completion, reduce resource requirements, but also just increase the likelihood of success when we can adapt through the conduct of the trial. There are different types of adaptive designs. So easy ones is when you do a blinded sample size re-estimation during the trial. That's, of course, an adaptive design, but a very easy one. In interim analyses for fertility are very easy ones, but then you can get also really into fairly complex ones with adaptive enrichment designs, adaptive dose selection designs, response adaptive randomization, all kinds of seamless design. So it can get depending on what you want and what you think you need to adapt, it can get really complex. Now, for this talk, we will discuss primarily 5 key principles of adaptive designs, and that is something I will do, and then Frank will go more into some of the related issues around adaptive designs. When you will go through the whole presentation, you will see that all these principles are actually not adaptive design specific, but probably valid for any kind of complex innovative trial design. So I will start with the first principle and that you need to justify why you do an adaptive design and why you don't go for a classical clinical trial without an adaptation during the trial. There's guidance already from ISH E8 and E9 that you should have a clinical, valid clinical question which should be answered with appropriate studies. And then E9 goes much more into details what that appropriate would mean. And all those criteria, of course, remain valid. And what you need to do when you think about doing adaptive design, it's very important that you do a sort of a benefit risk assessment where you say, what are really my advantages I could get and what are actually the risks associated? And there are with adaptive designs due to the fact that you run interim analyses, etc. There are intrinsic risks that the interim analysis could change the conduct of the trial, could introduce things you don't want to see at the end of the trial and which may be difficult to interpret. So the feasibility, validity, integrity, and flexibility adaptive designs offer, that needs to be balanced out with the intrinsic risks. So the advantages should clearly understood and they should outweigh the inherent risks. And those trade-offs you have to do by yourself before you decide on it. And it would be good when you approach regulators, when you're pretty open about your thoughts and not let them actually guesswork what you had in mind and why you propose this adaptive design. And sometimes it's obvious, sometimes it's not. Another really important principle is what you need to do in terms of planning adaptive designs. E9 already recommends a lot that you should plan key things like your primary analysis and population, etc., in the protocol. And of course, that's valid for adaptive designs, but you need to go probably a little bit one step further. You should start also with the estimate. That's the key thing. And then move on what you really need to do also in adaptive design to get to a sound study. Aspects you should really think about is the anticipated number and timings of interim analyses. That should be in the protocol, the rationale for and the type of the adaptation, the anticipated rule governing the adaptation decision, how you should get to that, if you have an adaptation committee and IDMC used for that. Then, of course, the statistical methods to use to make an adequate analysis, that should be pre-specified. Very important, the approaches to maintain trial integrity. That's around the confidentiality a lot during interim analysis. And then, of course, the approaches to maintain type 1 error at the end for confirmatory trials. And of course, different types of adaptive designs require different degrees of pre-specification. When, for example, you do a futility analysis, you just can write in that you do the futility analysis, and even the rule does not need and probably should not be outlined in the protocol. But when you do those adaptive dose finding trials, then you should also write in how you think you decide about your dose, for example. Another important aspect, which is more complex in adaptive trials than in normal trials, is limiting the chances of erroneous conclusions. And this impacts efficacy, but also safety. And you need to think really about both sides. For efficacy, the standard approach in confirmatory trials is really to limit type 1 error. At a pre-specified threshold, we know usually the 5% two-sided, and that may be, depending on what you do, that may be a fairly complex undertaking, and you should really present a sound method for that. When you use Bayesian designs, and this is something Frank will allude to later on, especially when you use Bayesian decision rules, that maintaining type 1 error could be tricky, and there needs to be more set. So that can become quite complex. Another thing which is important is the reliability of the estimation. The study is not only type 1 error control for the efficacy. That's in order to demonstrate that you have an efficacy advantage, but for the benefit risk assessment, we usually take the point estimate, and therefore the point estimate also should be reasonable. The rule is that you should have no or at least limited bias at the end of the trial, and a number of adaptive designs inflate or bias the estimate you have at the end. When you do an interim analysis for efficacy, to stop for efficacy, of course, when you stop for efficacy, you have a rather overestimation of your effect. I think that's well known. And when you have an adaptive design proposal, you should therefore really evaluate also the appropriateness of the bias and the validity of your estimates. It's an important part of what you should think of. And in some cases, the bias and the variability could be calculated analytically, and then you should do that. In others, you have to use simulations. And for some designs, you can actually correct for the bias. And then, of course, when you have some solution there, then you should use that solution as well. Another really key point is the maintenance of trial integrity. And we have to understand that adaptive designs are more complex in this regard than designs where you don't do an interim analysis. And the knowledge of interim results, whether individual or at treatment group level, has always the potential to introduce bias and influence the conduct of the trial. And one thing you should anticipate is that you should do an analysis after your interim versus before the interim. And when you see fundamental differences there, then you have a fundamental problem to explain. And that could be just by chance, but it could be also that something leaked through and that actually your interim analysis has influenced the conduct of the trial. And then it becomes quite difficult in terms of trial integrity. And one thing is, of course, that you have to limit as much as possible the information you gain during an interim analysis to a broader, to as small as possible audience. IDMCs are even more important in adaptive trial designs than in others. But you cannot avoid often that something leaks out because people know you do an interim analysis for fertility. And when the trial continues, they may start thinking about what you have seen and now everything is glorious. And they might start enrolling different patients than beforehand, and already you have an issue. So you have to be very careful with that kind of information. Good. With that, I hand over to Frank.
Thanks, Uli. So Uli presented some key principles that we should be aware of. And what I'm going to do is now to focus on a few special topics, 4 of them. starting with monitoring standards. And in many of our phase 3 trials, and I'm focusing on phase 3 trials now, of course, we have DMCs already set up to ensure the patient safety and help assure the scientific validity of the trial results. And by definition, DMCs are set up so they fully understand the trial, the motivations, the details of the monitoring and their own specific role. So it would be very natural for the DMC to take on this additional responsibility of recommending the adaptations according to the pre-specification in the protocol. Adaptations based on non-comparative data, such as blinded sample size re-estimation, typically would not require a DMC. And therefore, with that being said, on this slide, as a consequence in confirmatory trials, there should be the general rule that sponsors are not involved in any interim decision-making. They should not see any interim results. Exceptions are possible, but they should be pre-specified when such exceptions occur and how the communication will then take place. And maybe the last point is just the role of the independent analysis centers. They should ideally also be outside the sponsor's organization. So this is the visualization that you have in the middle, the data monitoring committee being fully independent. In the upper left, you see the data analysis center, which should also be external. And you may need additional experts to the DMC because the DMC is taking on additional responsibilities that may require specific expertise, for example, in terms of the statistical methodology. At the bottom, you see then ring-fence the sponsor organization. In order to preserve trial integrity, you should have this group of people being not on the DMC, not seeing the results unless really it is absolutely needed. And you should also pre-specify who those sponsor representatives are, ideally independent of the study team, senior officials. You should have firewalls in place and all this, what we already know. So these are a few comments on the DMCs. Moving on to the next topic, adaptive designs using Bayesian methods. So one could think about distinguishing between two types of adaptive designs using Bayesian methods, namely whether you use a frequentist inference at the end of the day or you use Bayesian inference. So if you use frequentist inference, classical example is if you base your fertility stopping rules on some Bayesian methods. So this would be one possibility where otherwise the final inference is fully frequentist. It's only for fertility stop could be used using, for example, Bayesian predictive powers. These designs otherwise would be fully frequentist, and therefore the standard requirements, the planning aspects and all this, that would be fairly standard with respect to the final analysis. Now, if you do use Bayesian inference for the final analysis, based essentially on posterior distributions of the targeted estimates, then examples come to mind like Bayesian group sequential designs using non-informative priors. They're probably fairly standard, but also you can be more advanced and use historic information, such as in pediatric drug development, where you want to use informative priors. And in those cases, probably you will have to have a few additional considerations that do not arise in designs that use frequentist inference. So this is what this slide is about. So we are talking about the construction of the prior distributions, this choice of the decision criteria, and understanding the operating characteristics. So with respect to the prior distributions, when you use weakly informative priors, this is probably uncontroversial. But I think there's a little typo in the second sub-bullet. It shouldn't be, it should read for informative prior distributions. It is, however, critical to absolutely document upfront the sources of the prior information, the relevance for the adaptive design, and strategies, also specifying upfront strategies to resolve any possible prior data conflict that you might see at the end of the trial. Decision criteria for trial success within adaptive designs should be pre-specified. This is also true for Bayesian inference, and thresholds should be selected such that success criteria are clinically meaningful and also meet common levels of success based on the chosen operating characteristics. And The operating characteristics, of course, we often will require, especially when using Bayesian methods, you will often require simulations, and we'll talk about simulations on the next slide. But when using informative priors, operating characteristics such as type 1 error rate would only be of interest in scenarios that are consistent with your prior. So these are all additional considerations that you will have to think about when embarking on adaptive designs with Bayesian method. Now, I already alluded to simulation studies in complex adaptive designs. There will be fairly routine use of simulations that we can all foresee. And this is with respect to several objectives. You could use them, for example, to demonstrate or at least to evaluate the type 1 error rate, also to look into the reliability of estimates, but also to identify good adaptation rules at the design stage. In most cases, simulations can only support type 1 error rate. So if you can never really strongly prove type 1 error rate using simulations, therefore, if analytical methods for type 1 error rate are available, they should be preferred. Now, importantly, simulation studies are, it's one type of experimental design. So we should plan and conduct a simulation study as rigorously as we actually plan and conduct a clinical trial. So you see at the bottom a long list of points to consider, because you should not just start simulating and then reporting lots and lots of outputs to different stakeholders, but you should really have a structured approach and really think about your simulation study as an experimental design. And so you need to be clear about the objectives, the operating characteristics, the design options, but also the current state of information to inform your scenarios, possibly your priors, the scenarios, that is the what-if scenarios that are not under your control, but also talk about the implementation, including the data generation process and how to document the simulations. So I received 1 minute ago that I had only 1 minute left. So I'm going to skip this slide just to say all the considerations are important in both early and late stage development. Although the relative weighting of the principles, they might be different in different stages of drug development. But in principle, we should always think about ahead, of course, about these and how your particular trial, whether it's phase 2 or phase 3, they would fulfill those principles. With this, I'm coming to a discussion, our discussion. So in a setting where an adaptive design can be justified and motivated, they can lead to a variety of advantages, but any adaptations for the purpose of our presentation, the way how we define adaptations, they should be pre-planned and carefully executed. There are a number of practical operational issues, of course, that need to be considered, and the relative importance of the different advantages and challenges will depend on the phase of development and, of course, the trade-offs between these advantages and the possible complexities that come with an adaptive design. Because any adaptive design will introduce additional uncertainty, and you need to make sure that this additional uncertainty introduced by an adaptive design, that you don't risk too much the 5 principles Uli was mentioning. And some topics probably need more discussions as a community, such as the use of Bayesian methods. And with that, I think I'm concluding. Thank you. And the next.
Thank you very much, Frank and Uli. Where are you? So we will take questions once all the 3 talks are done. And for those online, please remember to put your questions in the Q&A chat box and not in the chat itself. So it's now my pleasure to introduce the next 2 speakers. So it will be Wolfgang Jacquet, who is a biostatistics subject area coordinator for the Belgian Federal Agency for Medicines and Health Products. He is also a professor in biostatistics at the Vrije Universiteit of Brussels, at the Faculty of Medicine and Pharmacy. He is an active member of the methodology community of the EMA and a former member of the Biostatistical Working Party involved in the writing of the reflection paper on platform trials, which also makes that a very relevant speaker for this session. The other speaker you have already seen a few times. It is Benjamin Hoffner. who is the head of data science and methods of the Paul-Ehrlich Institute, which is one of the 2 national competent authorities in Germany. He is also a regulatory task lead in the recent IMI2 project EU Pearl on platform trials, and he is involved in the temporary drafting group on platform trials at the EMA. So welcome, please go ahead. Do you want to put it down first?
Yes. 1, 2, 3, yes. Okay, when testing, I've got too many slides, I've got a deal with Benjamin that is that if I am overrunning the information of EU Pearl, if you would be interested, is at the end, but I try to be polite and I'll try to be fast, as fast as I can be, without losing clarity. And I'm going to try to tell a story, and I'm going to try to tell a story to make clear what challenges we face with generating clarity for industry, but also clarity in our own heads with respect to complex trials. And to do that, we focus on platform trials. And platform trials that are a hot topic. We had platform trials in COVID-19. And the question and the reason why I stand here also is also to tell you why it is not so obvious to go from COVID-19 platform trials, which were extremely successful, by the way, with the drawbacks, but we succeeded in having a very fast evolution of treatments, repurposing of medicines in COVID-19 setting. But it is not completely obvious that we can repeat those successes and that approach in other indications, in other fields of clinical investigation. Now, to make that completely clear, I'm going back to the basis, and you all know that. Oh no, first disclaimer. Huh? Okay, good. I disclaimed. Voilà. Now the essence, and the essence is causality. What we try to do is to pinpoint a treatment under investigation as the cause of the observed improvement of the patient. We want to see the treatment effect. Now, causality is a difficult thing. Now, when considering a situation where you change only one aspect and consecutively other aspects change, changing one aspect is typically a treatment in our case, then we can attribute those changes to actually the treatment, to the intervention. Now, when we look at disease, we've got a problem. Circumstances of a person evolve over time. Treatment evolves, care evolves. The situation is never changed by only one aspect. Got always time. You've got always interactions. So this setting, this scientific setting, can't apply. So we have to do something else. And that something else is we will compare groups, or we will compare a group with itself in time. Now, how can we do that? Because then we even change more. It are different people. And we explicitly have time in there, and we know it evolves over time. So it's a bold move, and it works. It works. Why? 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 That is because of probability. We generate a probability experiment. The reason why I tell about probability experiment is that we must be well aware. Probability is introduced at different levels. First level is we've got the population. We always say that we will investigate a population for an indication. It's completely untrue. It's not true. We've got a population, and that population is governed, well, we can describe it probabilistically by all features and distributions and multivariate distributions those features have in the population. Good. What happens is, and what is more relevant, that is that we have a selection. We have an approach to our medical care. That approach to our medical care most often is reactive, okay? The patient has to go to a doctor and say, doctor, I've got a problem. And from there on, it goes further. So it is a probability distributions of features with a selection. That selection itself has probability features, or we model it as such, okay? Good. And then we add yet another layer to be able to do those comparisons, and that is the randomization, the attribution of a treatment. There you go. We are nearly there. And then if all that is good, we have a framework where, and that's mathematical statistics, probability in combination with decision theory, has a framework that is the basis for effect estimation, but also for having an uncertainty on the effect estimation and an uncertainty on the uncertainty. Good. We can then control for what? For the probability to mistakenly attribute causality. So it's different from our decision. If we say, you've got marketing authorization, actually, we decide there's causality in it, and the effect is large enough. Okay? That's what we do. Or that's how the outside world perceives what we do. I'll get back to that later on. So there you go. Randomized clinical trials, okay, with internal concurrent control, I've got to look there, are still considered the standard in trial designs. And then you've got it when appropriate and feasible. Good. And I would say that's the basis. And appropriate and feasible, I would like to change that, because not really, but an interpretation of the appropriateness. We've got a tsunami of novel possibilities, okay? We've got limited patients. We have got limited resources. So if we want to profit as fast as possible from that tsunami, we've got to have an interpretation of that appropriateness toward more technical designs. But we've got to be aware that we need to have effect estimations for what? For the system we have, for how our clinical system works. Is that reactive? Should that be different? So there we've got to have the right representing probabilities. we've got in our clinical trials to have something of a selection that mimics our healthcare system. The question, how do we have to change that healthcare system? That's no other ball game. We've got the system, we've got now, okay? So there you go. So we've got two aims, decision-making. Oh, blast, took the wrong. Oh, hop. See, synchronization is good. We've got there decision-making and precise estimation. And for that, really going forward and speeding up, we've got complex design, we've got artificial intelligence, we have real-world data. How to move forward? That's a question. What we've got guidances, we've got clinical trial regulation. What can we see? Well, first of all, those regulations, the ICH E9, clinical trial regulation, and others, clearly are frequentist inspired. But all of them leave open possibilities. Actually, they are well written. There are, in the sense that there is the intent with an opening to possibilities that could not have been foreseen at the time of writing. So in that sense. But are they helpful? Yes, I think they are, but they need to be adapted in some way, and adapted in the sense there need to be additions to them. And that's a process that is not so easy. And that's a process we are engaged in. Good. Now, to go back and get a bit focus in this long story, beautiful bow, I hope. Okay. What is a complex clinical trial? Not so easy. If you've got a law, you apply the law. We've got a law, clinical trial regulation. If you go and look at clinical trial regulation and you try to find out, you will not find anything about what a complex clinical trial could be. And therefore also, since platform trial is considered a complex clinical trial, and I will show you it is, and you all know I don't have to show you. There is no information on what a platform trial is, but there is information on what we can consider a trial. Well, a trial, and you will have my notes, and I already adapted the notes I created, and there are some typos in it, and so please read them carefully, but with consideration of errors. Well, at first, a clinical trial is a clinical study. So it is an investigation in relation to human beings. That doesn't say much. But then one aspect is the assignment of the subject to a particular therapeutic strategy is decided in advance. That's an element I want to take with me when going further or later on. Now, the core. Why the COVID-19 experience is not a good reference template for confirmatory platform trial designs. Well, first thing is the COVID-19, a combination of master protocols, well, different platform trials, by the way. You all know that. And you've got a combination of a master protocol inspired by World Health Organization, driven, and sub-protocols to complete the master protocol and have instantiations that can be considered classical clinical trials. Reason to do that was efficiency, comparability, uniformity, accessibility of sponsors to increase participation. So also sorry, speed up in the investigational process. That it was. But it was a unique situation. We had an extremely continuous flow of patients. It's a luxury. If you want to investigate something, at least you've got subjects, and you've got subjects everywhere. And due to the system installed, also sites that were not standard participating, sites participated. There was also an involvement of the public to get into trials. So there was a unique opportunity. There was a unique system. Now, that is in contrast when you propose platforms for small impacted populations. That's an all other ball game. Well, I think there it can be most fruitful. So there's a distinction to be made. Now, for me, at the end of the regulator, there was, strangely enough, most often clarity in what had to be considered a probabilistic experiment. And it was, so you had that master protocol, you had sub-protocol, boom, you considered both, and thank you, being really too slow. And it was really clear what I had to consider as a probabilistic experiment, because they were more simple than what comes to us in these days as from industry. Good. In these days, we often get a clinical trial application that looks like a development plan, with enormous amounts of dynamic elements. Okay, good. So, platform trials. What is a platform trial? An approach could be a trial platform. So the platform trial in itself is not trial, but you can consider that it would be a trial platform with trials as instantiations generated to sub-protocols. And that's actually what happened in COVID-19. You could see it like that. But it really differs from the wide variety of definitions we find in literature, but also in our documents are not uniform at present. Good. Well, here, that's a bit what we had in COVID-19 type of things. But what I want to show here, going very fast, that is actually, oh, blast, that was the wrong button. That here, you see here at week 12, you've got 3 studies, okay? And you've got 3 sites participating, but those sites participate in different studies. So one thing that made clear, thinking about platform trials, is that actually with regular trials, we don't have independence between trials. What we perceive as a problem with platform trials being that there is dependence. Well, actually, I think that is also a problem in ordinary trials. Going back further here, and I've got probably only 3 minutes left. So I'm going to talk fast about independence, independence, and independence. And I'm going to do that from abstractions of what I, in my regulatory practice, saw happening. And voila, that's one. And what do you say? You've got population, and suppose there's a natural influx, so that probability in selection is all right. And I've got control 2 treatments. And what we typically would say, oh, this is an ANOVA type of design, okay? Good. How to analyze? Well, no. No. It is not an ANOVA type. It's a platform type. That is submitted as a platform type. Okay, good. Well, we don't have to do any alpha control or anything at all because treatment 1 and treatment 2 are independent. Okay, good, independent. And then I don't get it completely. Are independent? Independent. Well, probabilistically, they are not. Because you've got 1 minute left. Thank you. It's not, I don't have to repeat it, but funny. Good. So actually, those are dependent. And we get a group effect. Namely, if by accident that control performs extremely bad or extremely good, we will in bulk accept treatments or in bulk reject treatments. So we've got to be aware, so there is probabilistic. Okay, good. Another example I had, that is in one clinical trial application, to say actually, we ask you that as one clinical trial, but it are 2 independent clinical trials. And there you go. There you go. Who is deciding? I've got a big population and a small subpopulation. Who is deciding in that population? Who is going to have the treatment if in the blue population, if the person that fulfills the requirements of the blue population will be included in the green trial or in the blue trial? Well, there, the clinician decides. Whoa, blast, that's a problem. If it would be random, I would have a problem because it's not the natural influx, okay? But it is at the trial site, the clinician decides. There, I've got the clinicians believe. So clinicians talk with each other, clinicians have conferences that we do. So independent. Independent, no guarantee on bias, 100%. We finally, since I don't have to take the decisions, it was decided that it was independent. But it raises problems in our current practice because I don't think that this is specifically a problem of platform trials. It is more general. It is due to the fact that we try to introduce complex trials, that we see that in our approach to trials, there are flaws, and we should address them. So we've got probability independence, independent modes of actions, different sponsors. And then you've got the corrections to correct for them. And then there's the question, how linear can a human being be? I'm counting buff. And that doesn't mean that I think that we shouldn't do corrections or transformation of corrections. All those corrections somewhere have linearity without interactions, by the way. You can't do that. Sample sizes will be too high. So that's the question, what can we accept? When is it dominating? When is it not interfering and helping? And where is the border in what we can do? Okay. Shouldn't we adapt decision making? I want to, I'm talking too fast now. I want to end with this because I think that we should move towards what is called most often with a strange name, pragmatic trials. If we have to move fast, we will make errors. There will be problems. We have to move fast, according to me. We have to guarantee safety. And we have to consider the decisions more and more as, for now, we've got indications to be confirmed that there is efficacy. And we've got estimations and we've got problems with those estimations, be honest. And we have to have randomization in clinical trials with more natural selection flows. And we have to correct if they are not natural. In my humble point of view, sorry, Benjamin, I did it. Fast forward. Ta-da, first slide. So
I
sorry.
think, Benjamin, if you would like, you can take a few minutes and.
I just grabbed the microphone, and I've talked more than enough yesterday, so I will be very, very quick. I just wanted to briefly introduce to those of you who haven't heard the EU Pearl project, which is now over. So it's all in the past, and it was a project on platform trials and with a huge consortium. So there were many, many different stakeholders involved, including the Paul-Ehrlich Institute and others. And there was one task which tried to deal with a clinical trial methodology, also operational and statistical and regulatory aspects. And this is where we were mainly involved. And what we tried to develop there was then somehow tested in 4 different disease areas with different issues. And we tried to develop platform trials for these disease areas. We didn't run the trials though. So it was not that these trials were really set up and run, but they might be run in the future. But what is good, all the efforts we made are available online. So there's the webpage eupearl.eu. You can go there, and there you will find, and I have it on the slides, different resources. You have tools, you have publications there, and there's still publications in the pipeline. So for example, there's one publication which we currently are about to submit about regulatory issues. And there's also a community where you can subscribe to if you're interested in platform trials. And hopefully, I haven't received any communication yet, but hopefully in the future, there will be communication on further things around platform trials to evolve. So you can go there, and it's everything in the slide deck. And yeah, with this, just a really, really brief overview that you also know that it's there. And we will also use these information and all we learned in the project for the draft guideline we are currently preparing. So it's not gone. So we will try to make this sustainable also from this angle. And yeah, I hope this is enough for today, and we have more in the discussion.
Thank you both for these nice presentations. It's my pleasure to introduce the speaker for the 3rd presentation in this session, Dieter Hering. He is Executive Director Biostatistics at Novartis, and he has a lot of experience in biostatistics. He has a main focus of interest in multiple sclerosis, and so he's an expert and will share with you a very challenging and interesting example of regulatory interaction and a quite adaptive design that was in the end accepted by regulators. So Dieter.
Hey, you've already heard I'm Dieter Hering. I'm a statistician working at Novartis. I want to share some experience with the regulatory interactions that we had based on the NEOS trial, which is a study in pediatric MS patients. And I should maybe move my slides first. No. Okay. I have been presenting the trial itself in a previous presentation here in this forum, so I'm only going to touch the design elements to a level that you can understand what it's all about. In the second part, I'm going to summarize the regulatory interactions we had with FDA and EMA. And then lastly, I tried to come to some kind of conclusion. What learnings did we have from this experience going to the complex innovative trial design pilot process also with the FDA? What are the learnings, and how could that be maybe digested into some kind of guidance for others who also want to have adaptive designs in the future? So I should stand here, then I can read. So first of all, pediatric MS is a rare disease. Most patients are adults, and only a few have an onset already in childhood. And for many who do have an onset in childhood, they only remember it after that they are diagnosed as adults. So these are not patients who are available for a clinical trial because they didn't know when they were children that they had MS already. Second thing is disease similarity. MS is pretty much similar throughout the age span. The biological processes, I mean, are similar. What is not similar is the quantitative contribution of these different biological processes to the disease. So children have a much more inflammatory disease course compared to adults that ceases over time with older age. So we need to take that into account, but basically we can model this. Then pediatric MS patients are a vulnerable population. What I mean by this is there's a risk of irreversible disability. MS patients, they lose brain volume, they lose neurons. That's most likely an irreversible process. And so we need to take care of these patients and hopefully try to not use inefficacious medication like placebo or low efficacy treatments. That's clearly a concern to the children and their families and the caretakers. Lastly, there's a high unmet need. So in adults, we have, in the meantime, about 20 approved medications. And in children, we have managed to test one medication successfully and approve it. There are other medications being used that have not been tested or that have failed actually in the trial and are still approved. But there is only one trial that has been completed and shown superiority over an active control and therefore was approved. So there's this imbalance between the situation in adults and pediatric patients, and that causes a lot of frustrations with both patients and physicians. Now, the NEOS trial, what's the NEOS trial? The NEOS trial is a 2-year double-blind triple dummy design in pediatric MS patients. It compares 2 new medications, ofatumumab and siponimod versus an active control, which is Gilenya. And Gilenya is the one medication that has been tested in pediatric patients and have shown that they can reduce relapse rates by 80% compared to interferons. And based on that, it's globally approved for use in pediatric patients. The primary endpoint is the analyzed relapse rates. That's a standard endpoint in clinical trials in MS. It just is a recurrent event endpoint. A clinical relapse is a period where a patient has neurological symptoms, acute neurological symptoms. The trial also features interim analysis for efficacy stopping, and that's at the time when the last patient completed 1 year of exposure. Okay, this is a quick summary. Now the question is, why are we doing these things? And I think that's the key thought here. Why are we not just doing a normal trial, a randomized control trial without adaptation, without complexity? Wouldn't that be better? I think it's really key if we want to have adaptive designs that we can explain why we are doing these things. Why are we doing the adaptations? So I mentioned on the left-hand side the clinical challenge, and then the innovative trial design feature in the middle and the impact it has. So it's a vulnerable population. We want to avoid the use of placebo or inefficacious or low efficacy drugs in this population because there is the risk of irreversible disability. So how can we do this? Well, because we have one medication now that has shown so much superiority, 80% reduction in relapse rates, that maybe we could show if the new medications are similar enough to this efficacious medication, maybe that's good enough to say, you know, we can also approve it. But how similar is similar enough? That's, of course, the choice of the non-inferiority margin that comes into play. And we are working in a rare population. So this margin should be tight enough to ensure that the new medication is clearly efficacious, but it can be wide enough to still make it a feasible trial. And so this provides us with the scientific rigor of a randomized controlled trial, showing that the new medication is efficacious while still being feasible. Typically, a non-inferiority trial requires a larger sample size, but here the purpose is not really to show bioequivalence to fingolimod. The purpose is to show that the new medications are efficacious. Then another challenge is the integration of prior knowledge. So before we go into a pediatric development, we always complete the adult phase 3 trials. So we have hundreds of patients that have been exposed, And in MS, that's typically an illness of young adults. So it means we have patients down to the age of 18. Pediatric setting starts at the age of 17. So they are not fundamentally different. The on mean average age is about 15 in these pediatric populations. These are not babies, these are not toddlers, these are typically adolescent patients. So that the span is not very wide, and we can actually use some Bayesian design to integrate this information. So we also understand how pediatric MS is different from adult MS. As I said, it's more inflammatory, but it's not a gradual step that happens at age 18. It's something that happens gradually, so we can model this. And so we have a Bayesian design that uses some model-based extrapolation from adult patients to pediatric patients, and then we summarize this information, also including, of course, the actual pediatric information in robust map priors. That's a way of a Bayesian design where we add to the Bayesian prior from the model some non-informative components, so that if there is data conflict with the new study data, then the Bayesian prior is basically discarded or has less weight. The last challenge is there are few therapeutic options. So we really have an interest to bring new medications, new tested medications to patients quickly as we can, within what is scientifically reasonable. So we have this adaptive design and an efficacy stopping rule so that we could bring this medication to patients as fast as possible. And so once all patients have completed 1 year of exposure, there's an interim analysis planned. So I hope I explained all these features and why they are there. Now, this was already the trial. So I'm going into the regulatory interactions, and I show you a main challenge for reaching global agreement for these trials, because we want to run these trials globally, right? So we sent almost the same proposal to FDA and EMA, and you can see the initial feedback here. So extrapolation, I mentioned, from adult patients to pediatric patients. EMA had no specific concerns, could follow the logic of what we outlined, and considered this potentially acceptable. FDA, on the other hand, said there are concerns about the extrapolation models relying on unverifiable assumptions. We had to do a lot of additional exploration of all other possible prognostic factors, disease-modifying factors in the study. But finally, it was accepted as a design feature. But you can see there were different opinions from the beginning. And we suggested this non-inferiority margin of 3. I'm not going to explain that in detail, but it comes out of the single pediatric patient, and it would ensure that the new treatments are better than an interferon. So EMA considered this margin large, but also deemed it acceptable in the context of the specific pediatric setting and the rareness of the disease. While FDA said it's too large, and especially it doesn't account for between trial variability. So if you had a new trial, maybe the result would be different, and you need to somehow take that into account in your non-inferiority margin and have some kind of discounting. So finally, they recommended, after a lot of simulations and so on, that we implement a margin of 2 in this trial, and that was then decided and integrated in this trial design. Now, after the start of the study, FDA sent us an unsolicited letter saying that now they would agree that the margin of 3 could be acceptable. So we went back to EMA because we thought maybe that's a concern, whether we could adjust the sample size to this new margin of 3, and that was deemed acceptable. But then FDA said, well, the adjustment of the sample size to the new non-inferiority margin is not automatic. So there is a need for further discussions probably on that point. But you can see that the feedback is not the same globally, and you have few opportunities to actually reach out to the regulators. There's no direct interactions typically, so that makes it very hard to come with these new trial features and integrate them in a global trial. Also, patient design, FDA said it may be useful, while EMA considered it not acceptable. The interim analysis is the same. FDA in this case said they endorsed this proposal from the beginning, while EMA was of the opinion it could not be accepted for a PIP. So it took a lot of simulations, a lot of explanations to actually make this a globally approvable design concept. And it is now, it's running, and it's running well. So what are my learnings from this, and what are the team's learnings from this? I want to summarize this and somehow generalize it into some kind of guidance that I would provide to anyone who wants to do this, and maybe also for considerations by those in agencies and so on. What are the problems from a sponsor's side? So everything is, of course, from a sponsor's view. And I'm also talking about other perspectives because I think this is really important. So my first learning from this is different parties in a clinical trial have super different views on the same clinical trial, and they want to optimize different features. So for instance, as a sponsor, we have a large interest to bring efficacious and safe medications as efficiently to patients as possible. So the efficiency is an important concept. We want to be scientific, but we also want to be efficient and bring medications to that. That's what we see as our duty or as our job. I think from a patient perspective, and I would include the physicians here, one thing physicians and patients want to optimize is they don't want to be exposed to undue risks of adverse events, but also not to low efficacy drug if there is actually a risk of irreversible disability. So low efficacy drugs are a concern in such a situation. And also, there's a lot of frustration in this clinical setting about the situation of having 20 approved medications and tested medications in adults, but only one medication in pediatric patients. So having access, give us access, find better solutions is clearly an ask from both the patients as well as the physicians in this indication. My experience from the regulatory side is that the main concern is to minimize the risk of erroneous decisions, which is well understandable. So power, is the study powered enough? Or could it be non-informative at the end? What's the risk that we approve a medication that's actually not efficacious? So type 1 error and type 2 error control. And of course, also sometimes these adaptive designs and complex designs may be perceived as a shortcut. So I think that regulators may want to avoid shortcuts, and maybe rightly so. There may also be a consideration of the fairness between competing sponsors. I hope so there is. And the alignment between global regulatory agencies, topic that I mentioned before. I think people are aware in the regulatory agencies of this being an issue, but I don't think it's a problem that has been solved. A second learning for me is innovation always breaks with established tradition. So if you follow the regulatory guideline, you will never be innovative. And so the question is, why do we deviate from the guideline? And why do we deviate from the standard? There needs to be a good reason. I think it's key that we can quantify and articulate that benefit. Why are we doing this? And the means to do this, I think, is simulations in many cases. Simulations to show what's actually the advantage. And they're often, and I think that's the reason why the regulatory feedback is so different from different agencies, there's subjective components in this. So for instance, how do you weigh the weight to patients or the burden to patients versus the sample size requirement of a study? There is no closed formula to do that. It's a judgment call. It depends on the clinical situation and so on. Also the weight of historical priors, historical data, how relevant are they? How much weight should be given to the priors? That's not something that at the moment we can sort out in a closed formula. Or when is type 1 error inflation acceptable? Frank and the team mentioned in a patient design that's not strictly controlled, but when is it acceptable? It's a bit subjective. And so simulations can help also to address the question, what if we did what you think is the best, then what would happen? And to understand these different things that different parties may want to optimize, I think is key to have a meaningful discussion. So the acceptability of innovative trial designs, I think, depends heavily on the clinical context. It's not a one-size-fits-all, and therefore the collaboration between statisticians and clinicians, both on the regulatory side as well as on the sponsor side, is even more important than in a normal regular trial. I give you one brief example what I mean. This is related to the patient design and type 1 error control. So these are simulations. On the x-axis, you have the analyzed relapse rate. So how frequently do patients experience relapses? We expect that to be at 0.15 as the main scenario. And you can see on the y-axis, the type 1 error, basically. And the line, the solid line, horizontal line, gives the nominal level. So if you had a frequentist design. So if relapse rates are high, it's easy to see whether the new treatments are non-inferior to fingolimod because you can really judge that. The issue is when relapse rates are really, really low. When no one has neurological symptoms, then it's impossible to tell if the new treatments are actually better than fingolimod. There's a risk that you actually conclude maybe ofatumumab is better than fingolimod, but it may not be true. So the risk is when relapses are really low. Now, the clinical context comes here. Historically, we have done a systematic literature review and the meta-analysis. Relapse rates were never low in pediatric patients. There's not a single study that would report low relapse rates. They're always high. So from that perspective, if in the new trial where everyone gets active medications and relapses do not occur, you still know that new treatments are efficacious. So in this case, type 1 error inflation may be acceptable in the specific clinical setting. And I think that's true for many of these adaptive features, that there's a specific setting under which they may be acceptable, and that needs to be understood and explained. Now, a fourth lesson for me is we have means to understand the operating characteristics of a study before we do it, and then we can also understand what happened after the fact. And so the means to do this before you do the study is simulations. So simulations can be used to understand what would have happened if I used the FDA's best proposal or the EMA's or anyone's, and we can compare what are the features of this, where do we have advantages and where do we have disadvantages. I think it's important not only to cover the per protocol set, our preferred assumptions or the best assumptions, but actually the whole range of possible parameters, at least the plausible ones, not all the possible ones, but discuss the plausible ones, or do all, but then discuss it in light of the plausibility of these scenarios. After we conduct the study, we should have some tipping point analysis, and I think the key ones should be predefined. For instance, how much weight do we give to the patient price? So we can range this and say, what would have happened if I didn't have used the patient price and it was a main frequentist design? Would the results have been similar? Would the conclusions remain robust? So this is my conclusion, and I instead of having a real conclusion, I try to come up with something like a guideline. That's a bit ambitious, so forgive me for that. But I mentioned as the first thing, different parties have different views, and I try to come up with something that hopefully everyone can agree. So I think sponsors, regulators, patients all want that we test the efficacy and safety of a new drug as efficiently as possible and with as low burden to patients, without loss of scientific rigor, to make new therapeutic options available to patients. Now, I underline certain words, and I think any kind of adaptation tries to optimize one of these different words. It may be related to lowering the patient burden, or it may be related to maximizing the efficiency of the trial or the safety evaluations. But whenever you modify one of these, typically there are effects on the other ones. I think that's important to understand and to articulate what these advantages and disadvantages to have the whole context and to make it acceptable to all parties. Now, there's 3 sub-bullet points. My first one is make it understandable why you want to have these adaptive features. What is the advantage? And I think adaptive design features and complex innovative trial design should only be acceptable if they have objective advantages. But under these conditions, they should be accepted. So it's important that we can spell this out and have this documented for all parties. The second thing is we should understand the operating characteristics and limit the risk of erroneous decisions at the design state. We can do this based on simulations, as I mentioned, under the different scenarios. Topics are power, type 1 error, bias. In terms of extrapolation, I should not have bolded extrapolation. I should have bolded disease similarity, because disease similarity, when you extrapolate from a source population to a target population, the question is how relevant is that data? And so really the bolded word here should be disease similarity. And the third thing is, I think if you want to be successful in the long term with these innovative complex designs, we need to make sure that the regulatory decisions are understandable and well documented, that people can really follow it. That it doesn't seem random which product gets approved and which doesn't, and which design does get approved and so on. That we have a good documentation, that it's explainable and supportable by all stakeholders what the decisions are. And so I think key features here are that there is a clinical as well as a statistical success criterion predefined in the protocol, and that we have tipping point analysis so that people who disagreed with from the beginning with some design features can understand how the design or how the conclusion would have been if they had their optimal setting. I think this is key to really have acceptable decisions there. I hope this was a bit helpful. That's the end of my presentation. Thanks a lot for listening to me.
Thank you to all the speakers for some excellent presentations. Are there questions from the audience or online?
Maybe then we start with a, oh, you can start. I would like to also to ask the speakers to please come forward.
Thank you for the excellent presentations. I have a brief, Klaus Eckhardt, consultant and former regulatory or regulator. I have a brief question to the last presentation. I think the FDA is increasingly trying to include especially the adolescents already into the adult drug development programs and clinical trials. Was this discussed, or what was the reason why this was, at least for the new drugs, not considered? Thank you.
It is, it has not been discussed in this specific setting, but I'm aware of discussions ongoing on new trials for exactly that purpose. It's certainly still the standard that we complete the phase 3 program and have results before we start the pediatric program. But these considerations are there, especially since the age difference is not huge. So it's typically 16, 17-year-old patients or so, pediatric patients, I mean.
I also want to thank you, Dieter, for the excellent presentation, and would like to ask a question about efficiency and speed, because we are talking about this a lot. How long did it take you to convince both authorities to go for the solution you proposed?
Yeah, okay. I think it was about, it was probably about 3 years in total. So we started in 2016, 17 with these discussions. It's clearly, it's a process. One of the things is the FDA process of this complex innovative trial pilot process has a very strict follow-up. So basically, I think you need to submit the simulation results 3 months prior to a meeting, and then there's the meeting, and then there's, so it does take time just from that perspective. And there's also a lot of work that needs to be done. Yeah. So it really, you need to know why you're doing this if you want to start it.
I also have a question to you. I'm not sure as a panelist, but I think it was really interesting to see what you did. For me, it was a bit, I'm not sure. When I was looking at your type 1 error rates, in the scenarios where you don't inflate type 1 error, I would assume that also the gain in power is rather minimal. But this was the most likely scenario you had. So perhaps you can comment on this. And the second question I would have is, how do you make sure to communicate your simulations in a good way? Because I think this is really crucial for us also to understand how sensitive your simulations are to specific choices. I mean, me as a statistician, I would love to have code, and I can play with the code to understand whether your choices were sensible, and also to see how strongly they are affected by perturbations to your assumptions in a meaningful way, so that we can have a better informed discussion and not only have to rely then also on this one table with operating characteristics, which is not perhaps useful to clinicians or to statisticians and the agencies.
Okay, I think there was more than one question, if I can correct. But so I start at the end. The information of the simulation is not public, but it's part of the CID process, so the pilot process with FDA, to hand in the data as well as the programs. So I think on the FDA side, there's a review, there's they agree with the characteristics. That's the information that's probably available. You can also see the publications from both the Novartis side as well as from FDA side on this particular trial design and what they think and what the concerns are and so on. The other part, so I think specifically with these robust priors, there is the issue that, well, the advantage of the robust priors is that if there's data conflict, the prior has less weight, and there's less, it doesn't bias the result much if there's discrepancy between the prior data and the new data. So that's a good thing, but it also means that you don't really know a priori how much weight this patient prior carries. And therefore, if there is data conflict, you may actually run into an underpowered study. The way we try to take care of this is that we have a very reasonable probability to stop at the interim analysis if it's the protocol conditions. And if actually, there is a data conflict, then the study will run for the full 2 years, and then we should still have an informative trial. Hope that addresses some of the concerns, yeah.
Can I have a brief follow-up question? Is that sorry? Is the microphone working? Yeah. I mean, you mentioned how you submitted the code and your data to FDA, but was there also a process to submit the code to EMA? Because well, this is where I am working, and I'm wondering whether we should establish also a procedure to provide code at the EMA, because I think it's really useful and sensible to have an informed discussion up front and not only have to rely on a briefing book. And this is a question also to us.
Well, I answer it well. From our perspective, at least, my experience is that FDA has access to our data. Also, when we have a drug submission or so, we submit the files and the programs and everything. They do their own analysis and come up with their own conclusions. While with EMA, my experience is that we get requests and then we do the analysis and have the results, but not the programs and not necessarily the data. That would be my understanding. It's also the case for this.
Yeah. Thanks a lot for the very interesting presentation from principle to example to some philosophy on what we're actually trying to achieve with good clinical trials. Just quickly on the last question, I think the thinking about the raw data pilot that was presented yesterday, very much the thinking at the EMA side is to do stuff on top of just checking what the sponsor did. I think typically the complex trials are an area where we think access to data and analysis programs is going to be useful to better understand what's happening rather than for regular clinical trials. So I think it kind of matches. I had one short remark. I think I liked your summary on this shared objective, but I think it actually also transpires where our biggest worry is, because you formulated this as without loss of scientific rigor. Well, if you're neutral, you would have formulated as with scientific rigor. So I think we kind of share that we are at risk of losing some scientific rigor there. And I think it's really good to acknowledge that. Now, my question, lots of evaluating these complex designs is aimed at the primary endpoint and at its efficacy. But that's not the decision. That's just having a hurdle to decide, am I looking at an effective treatment or not? Because if I'm not looking at an effective treatment, I can kind of skip benefit risk assessment. Any thoughts on the panel on how this impacts benefit risk assessment more in general? If we have complex designs with adaptive decision making, but it's not the only endpoint I want to look at. And I also want to look at safety, and all of that is also impacted by decision making that either drops arms, changes populations. So any reflections there, what we need to do to make sure that we also understand what happens outside of the primary endpoint.
Very challenging question.
I think is discussing here for the specific example here, the pediatric study, right? There are few patients, these are small studies, and the phase 3 in adults are usually in hundreds or even in thousands of patients. And as I mentioned, we typically have patients down to the age of 18. So I think you would need to see extraordinary data that it would actually change the benefit risk of whatever you see in the pediatric setting. And I think it cannot really be assessed in the study. It's more realistic that you assess this on the market.
Yeah, I think generally I would argue a little bit against that and say you need to make sure in the beginning, before you make your before you start the trial, not only look at type 1 error rate, but also at the bias in your estimation. And the simulations you have to confirm is really also on that and the data prior issue with Bayesian designs. You need also on the estimate, you need to take that up front. I think at the end, we have to assume that sort of the efficacy test is an entry criteria, but the final decision is a benefit risk, and that is based on the estimation. It may not be only on the primary, certainly on the safety, but also on key secondary.
I would like to add a question because I had a very similar thought and it's related. If you have adaptations in your trial, I think it could happen that the S-demand also changes through the adaptation. So any thoughts on that? Should we refrain from doing adaptations that have an impact on the S-demand? Like you stated, in a platform trial, the control could change. The population could change. So this affects the estimate in a very direct way. And any thoughts on what we should do about that?
Just like to say that as with any adaptation, particularly when the estimate attributes are affected, like the population, the treatment, you need to define, like you would do with any, can you hear me now? Like you would do it in a standard trial, you need to define your estimate prior to implementing the adaptations.
And I would just add, I wouldn't call this a change of the estimate because for example, you're selecting a treatment or maybe you're selecting a population. So you're selecting pre-specified estimate. You're not changing the estimates. If you're truly changing the estimates, I don't know how you would interpret the combined data from stage 1 and stage 2. So I think we're talking about mostly about estimate selection designs.
I think there is of course a chance that you change your estimate, that with an interim analysis, you change your population by chance, but that is unwanted. And that's one of the risks associated with it.
I have a new questions with other reflections. I'm going to complicate matters a little bit more. And this is from real life experience of complex trials. So what we've seen, if you have adaptations in Bayesian design, but it doesn't necessarily have to be Bayesian, which depend on, let's say, a fairly long list of patient characteristics of things that happen in the trial, but you find out that your data was not correct. And you find out after the adaptation. Any reflections there on how we can assess whether that has a small impact, hopefully, or whether that has a large impact? it happened, and we didn't have an answer.
Just Kid asking back, do you mean that when in a dose selective adaptive design that I choose the wrong dose to move forward just by chance or by an error?
Decision rule can also depend on patient characteristics or outcomes that you've observed in the meantime. And you can actually have a wrong recording. There is an error in your database. And you do that adaptively along the trial. So you do not do that at the moment that you're 100% sure that you've cleaned all the data.
Like in response adaptive randomization.
Yeah, response adaptive, for instance. And yeah, we've seen it happen. There are mistakes in the data, and then the question for us becomes, what's the impact? Do we need to worry, or don't we need to worry at all because it doesn't have a huge impact on what the trial will tell us in the end? Sorry.
So the question from over here. Oh, sorry, Frank, you carry on.
An attempt to answer what is now specific to an adaptive design. I'm not fully getting it because you can also have errors in a non-adaptive setting, and also in a group sequential design. In a group sequential design, you could also have errors and you make wrong decisions. I'm not yet fully understanding the question, to be honest.
We need more microphones.
There's one more. Don't want to go too fully in the detail, but you might actually have made the wrong adaptation.
For example, also in a group sequential design, you should.
That might.
So that's it's a general.
Yeah, but group sequential is the, let's say, the data set that you use to actually make that decision is a lot smaller than we see happening in some of the more advanced proposals. So in that sense, the likelihood of occurring is a bit larger, and the primary endpoint is more often correct than other data.
The way, Kit, I understand it is that you argue that in an adaptive design, such things could happen with a higher probability. And that would be, for me, then something you have to take into account in the beginning before you decide on an adaptive design. That's one of these trade-offs you need to do in the beginning. The more complex your trial design is, the more likely it is that something goes wrong. And that you could screw up some things. There could be easy things where you think you can repair it at the end, and there could be things you can't repair anymore because you just screwed it up. And that is something where you have to evaluate in the beginning if the advantages you gain, if it's worth that additional risk. Sort of going down in the
I fully
right
agree,
direction.
but when it happens, it's also a statistical problem. When it happens, are you able to actually assess the impact?
All right, I think there was a question over here.
Thanks. A question to Dieter. So thank you, great presentation. And part of the presentation, you alluded to the fact that the lack of direct interactions with maybe the FDA or EMA was a problem, and that slowed up. And this is, as you said, now it took 3 years, which is obviously the system has failed these pediatric MS patients if it takes 3 years. So should we be advocating for complex, innovative designs that direct face-to-face meetings happen? Would that have helped in this case?
I think both agencies made an effort there. So we did have, as part of the complex innovative trial design pilot, you have 2 face-to-face meetings with FDA. So that's good. But of course, it doesn't involve EMA. There was a participation by phone from one agency to the other when we had one discussion so that they could at least listen in and see what has been done. So there were efforts made, definitely, from all sides. I think it's a good example, but I think this could be formalized a little bit more. From my personal perspective, is for these adaptive designs, it would be great if there was one kind of lead contact in the agency, you know, is aware of the files and and can be contacted also for clarifying questions and so in the meantime. That would be optimal, and that could also be used for interagency discussion, but I'm talking as a sponsor.
Quick response, or, no, it looked like.
Turn down the opportunity. So, I mean, maybe is there any reaction from the EMA on this, and whether they think more face-to-face interactions would be good?
I can't speak on behalf of the EMA, I'm sorry. I think it's useful to have these interactions. There's the cluster meetings, but I'm not sure whether they are in a timely manner feasible to really discuss these matters, but at least they could be helpful, but I cannot speak on behalf of EMA.
Then
I think
we have a
there's question
few
up here.
online questions as well.
Maybe
so
one
let's take
question here,
one quick question and then go to the online questions.
Thanks. I have one question for Wolfgang. I'm wondering, how would you define validity in platform trials, and what comparisons are protected by randomization? Are you hearing me okay? Okay,
Sorry.
I will speak slowly.
Maybe I can repeat, I guess.
Yeah, sure, you can repeat.
How do you define validity in a platform trial? And the second part was, how do you...
Which parts are of the comparisons are protected by randomization? How do you make that happen?
Well, what is protected by randomization is actually some answer whether or not there is an effect, and only in one way. If it is significant, there is an effect. Good. An effect for what? For actually also the selection process. So that means that if you have in practice the same selection process on the same population, then there is an effect. It's a conditional. And there goes your answer about the validity of the decision and also of the estimation of the effect. It is only valid if that selection process is somehow related to reality. That means that also here, I think there is something that needs to be done to go outside of the trial. And that outside of the trial is try to match the selection process of the trial with reality. That means that we have to try to get a canvas of reality. I don't think that would be the responsibility of one sponsor only, but that would be the responsibility of some kind of more societal thing where everybody has to benefit, so should contribute in some way to do it. That would also give the opportunity to see how we can reach more people with a medical problem, which we don't necessarily do now. So the validity is the conditional element of the trial.
I have several questions from the chat, but I would like to ask two, and maybe start with the first one. Your colleague from BFAM, Jörg Zinserling, has asked that there was a lot of emphasis on the importance of simulations to understand operational characteristics. And at the same time, plausible scenarios can become very complex. Is there an agreement that trial characteristics like censoring patterns or dropout patterns need to be considered?
I think all characteristics need to be considered when you are looking at the operational characteristics. Anything that can influence the trial results has to be considered. So I think that some transparency about that is needed when submitting the simulation studies.
You have to make a convincing case that you describe the operating characteristics sufficiently well And that's the difference to an analytical method where you know for every situation what you do, and when you do the simulation, you just can't select a few scenarios. You have to make sure that you get a holistic view of your parameter space and a reflection of that in your simulations. That could be tricky, of course.
Second one is a bit different. It's more to you, Uli. You said that there should be a lot of caution about trial integrity when doing a futility or interim analysis. And I have one question for that. Futility analyses are usually pre-specified in the protocol, sorry, including relative time points. Thus, it can't be really avoided that investigators are aware that the trial goes on beyond this point. Would the conclusion be that futility analyses are not recommended?
No, it's not that. It's just that you have to be aware of the risks and that you have to mitigate the risks as much as possible. So, for example, never write the bar you use for the futility analysis in the protocol, or keep that as confidential as possible, and limit the amount of information on whatever, not only futility, but whatever you do outside, because other people may be able to use that information and to recalculate probability of success and say, okay, this goes in the wrong or in the right direction. And immediately you could have an impact on the trial conduct. And that's something you have to avoid.
We have 2 minutes left. Are there more questions online or in the audience here?
I have one more.
Go for it.
Oh, no? Okay. Wolfgang, I have a question for you. Appreciating your comment that we should move fast and will do mistakes while not compromising on patient safety. Could you share which mistakes are most frequent and avoidable in platform trials? For example, what should a sponsor avoid?
Well, I don't do statistics on email level. So what are the most frequent mistakes? It's perhaps Kit, you I was no, it's very hard to answer that, isn't it? That in this stage, what kind of mistakes in platform trials? What I see is that a lot of things are being proposed, and a lot of things are extremely complex. So in what is proposed, it's a platform with elements of dynamic elements with no view and no information on trial size, competing trials. So what are mistakes? We are not there yet in trying to make statistics and have a clear view on all what goes wrong. We don't have the data. We are just organizing ourselves.
Just to briefly add, I mean, this is true in my view to all of the adaptive designs we were discussing. I think Uli captured it nicely in the talk in the beginning, that the question is, what do we gain from a trial, and what do we invest in the trial on levels of complexity, uncertainty, and so on. And I'm sometimes a bit, I consider it a bit challenging. We have a very, very adaptive design and we consider it confirmatory. Then I'm a bit struggling, to be honest. And there are situations where I can accept this, but there's many situations where I don't accept that, or I wouldn't accept that hugely adaptive design to be confirmatory, because it rather shows that we don't know that much in the beginning, because we need to adapt that much. And the question is also the rationale for why do we do specific adaptations, and what kind of adaptations are these? So a group sequential design, this is well understood, I think, and this is also often understandable from an operational perspective why we do this. But for other adaptations, it might be much more challenging on why we do this, and whether this trial can still be considered confirmatory. And I think this is especially true for platform trials, where we have so many complexities. We have response adaptive randomizations and so on. I think these are all valuable parts, and these trials can be really nice. But the question is, what is the purpose of the trial then? Is it an exploratory trial and will we confirm and replicate the results? Then it's all fine. Will it be the standalone trial for a filing? Then I think this is a mistake. The problem is we cannot preempt this because these trials then will be filed. So this is a bit the problem we have.
Thank you. I think this is a very nice closing statement actually for this session. Nice wrap up. So thanks to all the contributors for great presentations, great slides, great discussion. Thanks to the audience for very nice questions. And please enjoy the coffee break.